Hello, my name is Shengda, and I will introduce you a little bit knowledge about the microscopy and the cell imaging today. Yeah. So if you ask me or any microbiologists, especially the bacteriologists, what is their favorite moment? Probably their answer will be like they prefer to sit in a very dark room and look through a microscope and communicate with their little bugs. Well, it seems a little bit odd, but I have to say that the bacteria looks very gorgeous under some microscope. So in this lecture, I would like to show you the basic knowledge and techniques of microscope and bacterial image. So here's the lecture outline. So at the very beginning, I will introduce a little bit about the microscope, and thereafter is the light microscopes. We will get into details in the three types of microscope: the bright field microscopes, phase contrast microscopes, and fluorescence microscope. And as the last part, I will introduce you a little bit about electron microscopes. So here's the learning objectives. So after this lecture, you are expected to be able to use a phase contrast microscope to observe samples. Know the basic light paths, work theory of the light microscopes, know the mechanical component of a light microscope, a phase contrast microscope, and a fluorescence microscope. You will be able to apply the knowledge of the phase contrast microscope into practice and know the fundamental knowledge and types of electron microscopes, and know the differences, applications of light microscope and electron microscope. So at the very beginning, I would like to introduce you a little bit about two definitions. So the first is, what is a microscope? A microscope is the instruments used to see the objects that are too small for the naked eyes. While the science of investigating small objects using such instruments is called microscopy. Well, all the stories actually happens from an English natural philosopher named Robert Hooke. So he invented some one-lens instruments, and he named it as micrographia. And he used these instruments try to observe everything he can find. And as you can see from this photo, he observed some tree leaves, and he sees these small particles, small rooms under the instruments, and he gave the name cell to these little particles. And thereafter, people use cells to name the little component that from this our living world. Yeah. So a few years after, another Dutch people named Antony van Leeuwenhoek, who developed a new instrument, as you can see here. It is actually quite tiny, the size almost like your hand. It's very easy to carry these instruments and go to the nature, try to find some samples, and observe through the single lens here. So he used these instruments and try to observe, the fresh water, what's inside the fresh water. And also, he tried to observe, scraping something from the teeth, and try to observe with this microscope. So as you already know from the last lecture, the size of bacteria in the nature or in your teeth is around maybe 1 micrometer. And I have to say that this microscope, the resolution is 0.7 micrometer. So at this point, he actually observed the bacteria in the fresh water and also from the teeth. So Leeuwenhoek tried to give a name to these little bugs he saw, and the name is animalculus. and which means the small animals. Because of this, he was called one of the fathers of microbiology. So without the development of microscope, we couldn't really know further about the bacteria or microbiology. So here shows you what's happened for the modern microscopes. The modern microscopes is not one lens microscope. So normally they have several multiple lenses in the system. So we call it a compound microscope. As you can see in this photo, there's 3 different microscope. So for the light microscope, from the name you can see that they use light as the source to see through the specimen, or we call it a sample. So you can see that the condenser for the light microscope is glass well. But for the electron microscope is here, either both the transmission electron microscope or the scanning microscope, they use different condenser lens than the light microscope. So instead of use glassware, they use some electrical field or magnetic field, because they use electron as the source for all these instruments. So this is the difference between different microscopes. And we will use the light microscope as a sample to show that when you look through this your sample, because the light passes through this specimen, and then it goes a little bit like a cross through this eyepiece lens. So that's why when you look through the eyepieces, what you observed, actually it's a flipped image. So it's upside down. So when you're trying to move a little bit up or towards to your body, the sample, you need to be aware that you need to turn the other way. So the 2 important parameters that to define if a good microscope or not is the magnification and the resolution. So in the next slides, you can see the different magnification of the different instruments. So you can see that with your eyes can see the size or the resolution between 1 meter or more than 1 meters, and can small down to the 10 to the 4th meters. While to a light microscope, it can recognize a range of this from the 10 to the 3rd until 10 to the 7th. And they have a little bit overlap between the light microscope and electron microscope. But since electron microscope is much more powerful, they can recognize even the small virus and the structure of DNA, and even some moleculars or chemicals, and as small as an atom. So it's a very powerful instrument. So should we stop? Okay. So in the next slides, I will introduce you a little bit about the light optical microscopes. So light means, optical also means light. So you can use both. For a light microscope, it can be used to observe a living cell. For example, if you want to see the cell division process or the swimming behavior, as you can see from you were showed from the last lecture, you can use the light microscopes. And here this photo shows you some the mechanical component of a light microscope, or basic structure, a basic component of microscopes. So from the bottom to the up, you have this base, you have this light source, which gives all the light start from here. You have this screw that can adjust the focus. Basically, it's move this stage up or down, try to have the right focus. And you have this condenser that could focus the light from the light source and have it as a spot on your sample. And the light goes through your sample and will be collected by the objective lenses here. I will show you in a few seconds a real microscope. And then the light goes through this inside and will be seen by the eyepieces. So the very important mechanical component in the microscope is shown here: condenser, objectives, and eyepieces, and which carrying the lenses. So here I will show you the real microscope we have. So first we need to switch on with the bottom here. We need to switch on this microscope. And I will show you the light. If we just switch a bit. So here show you where the light comes from. This is the light source. And you turn a little bit here, can see there's a strong light here. If I block it with a paper, yes, you can see that the light goes through here. And then it goes to the sample or specimen, and then it goes here, and then it goes out from the, you can see here's a paper, through the eyepieces. Yes? So here's a light system for the light microscope. Yes? Just switch it off. And in the next, I will show you some objectives specification. So if you can see here, there's a lot of nodes on the surface of objectives. So how could you rate it? For example, there's information about which company it comes from. There's also information of magnification. So how larger they can increase with these objectives. For example, here you have the 10, it means it increased 10 times in size. And also you have this 5 or 4, you have the 20, and you have a higher magnification with 40, 50, or 100. So this could be distinguished by this color ring. So for each different magnification size, you will find this marked with different rings. And so they also give the information of like resolution. It's like with different resolution, you will have different, like the clear image or not. And here shows you some information that you can see. You can, from this line, you can see it's a flight field, or it's the aberration correction, or here shows you the magnification and the numerical apertures. Yes, and the important thing is you can also find some actual information with like pH 1 or pH 2 or pH 3. It means there's actual phase range inside these objectives. So you really need to change a little bit the sense on the condenser and then try to have it aligned with the objectives. So here shows you the working distance. Its working distance means when you have it focused with the eyepieces, the distance between the objectives and your sample. So it could be different. It could be different on different objectives. But here, if you see here, it's this site, or also the site that you will use in your exercise 3. It has the same working distance. So if you can get focus under the low magnificent objectives, for example, 10 times or 20 times, you don't need to change. You don't need to change the focus when you try to switch from one objective to the other. So it's much more easier to be handled. And here I show you a real objectives here. So if you can see there's information on this objectives here. The blue line, it means the light blue line means 40 times more magnificent. And what else? When you try to change a little bit, change objectives, you can just turn this, and it will change the objectives. Yes. Yes. And of course, it is the basic theory of some optical microscopes. And in these slides, you will find that there's a lot of different kind of optical microscopes or light microscopes. So there's more than this, at least only few. But in this lecture, I will introduce details only on 3 of them. So the first one is the bright field microscopes. So basically, it's the first kind of microscope that we used for 100 years before the phase contrast microscope was invented. So what is a bright field microscope? So it is basically, as I said before, it's the simplest because there's no any modification on the light. As I told you before, the light goes through the light source. With the condenser, it focuses on your sample. And then the sample will reflect the cells and could be refocused again by this objective lens. And it goes through the lens in the eyepieces and finally fall down at the end of your eye, and then you can see the image of your sample. So during this light pass, there's no any changes. So it just goes as nature. So what you can use with this bright microscope, you can use it to observe some staining fixed samples. For example, here you have bacteria, gram-positive and gram-negative bacteria, that you can easily see the differences with different color. And here with some staining method to the flagella, you can see this peritragus flagella of E. coli. Okay, just before we enter the phase contrast microscope, there's a problem with the microphone. So I would like to introduce again the slides and show you why we need a phase contrast microscope. So here shows you a photo of E. coli, the same bacteria as the other 2 slides, but without staining. You can see that you couldn't really distinguish the cells from background without staining. So this is because the low contrast of the light, the bright field microscope, because as I told you before, the light without any modification when they go through this instrument. And another disadvantage of this bright field microscope is the magnification is only up to 1300 times. So there's a low apparent optical resolution caused by this problem. So how could we increase the contrast of one sample? There's, of course, goes to the next microscope. We call it phase contrast microscope.